Okay, hello and welcome to this Twist Bioscience webinar, ensuring high-resolution CNV detection. My name's Chris Thorne and I am Senior Manager for Field Marketing here at Twist. So before we begin, I'd just like to do a little bit of housekeeping. As you've noticed, this is a webinar. All lines are muted during the webinar, but there will be time at the end for questions. And so if I can ask you to place your questions in the Q&A box rather than using the chat, and we'll get to those at the end. Following the presentation, if you have 1 minute to take a brief survey, we would really appreciate your feedback. In fact, this has been one of our most subscribed webinars to date, and we'd love to hear why you signed up and whether ultimately you learned what you were hoping to. So please do take a moment to give us some feedback in our survey. And so, in fact, it gives me great pleasure to introduce our speakers today. Joining us today from SOPHiA Genetics, we have Dr. Emily Paul, and from Twist Bioscience, we have Mark Consiger. So Emily is a molecular biologist with a specialty in genetics, having obtained her PhD at the State University of New York in Albany, after which she was a fellow at the Wadsworth Center at the New York State Department of Health, the Boston Children's Hospital, Harvard Medical School, and the Dana-Farber Cancer Institute. She joined SOPHiA Genetics in 2017 as a subject matter expert and is now director of the subject matter experts team for North America. Emily is joined today by Mark Consiger, who is the NGS Field Applications Manager for North America at Twist Bioscience. Mark has more than 20 years of academic, clinical, diagnostic, and translational research experience in inherited and somatic genetics, with 10 years of NGS experience in whole genome sequencing, whole exome sequencing, target enrichment, and transcriptomics. So Mark's going to kick things off today, and then he's going to hand over to Emily. So Mark, I will hand over to you at this point and let you take it from here.
Thank you, Chris. I appreciate it. Good afternoon, good evening, good morning to all of you out there, wherever you happen to be. My name is Mark Consiger, and I'm with Twist Bioscience. So as a prelude, what I would like to do is just kind of start talking about our technology, and that will be, I think, a strong foundation for the discussions that Dr. Paul will have afterwards. So let's just kick it off right here. So when I speak with people that are familiar with Twist or have a good idea of what we do, or maybe they're not so familiar, I pretty much get three main questions. What do we do? How do we do it? And why are we better? So what do we do? Well, very simply, we are a DNA synthesis company. But how we make our DNA is different than our competitors. So our way of making DNA is a precise method using microfluidics that results in a very high scaled methodology that results in high throughput oligos at very large quantities in a very short amount of time. These are our standing offerings currently right now. The first three are our foundational offerings in our SynBio product line: genes, oligo pools, and libraries. We have also been doing some very good foundational work and forward-thinking work with data storage using DNA. And as you may know, recently we've started to enter into the biopharma portion of the market. However, what I'm going to talk to you today about is our NGS offerings. So how do we do it? Well, the way that we synthesize our oligos is using standard phosphoamidite chemistry, but the platform that we do that in is different from maybe some of the more mundane or standard ways that you would think of. So, for example, rather than using a 96-well plate and synthesizing one oligo per well and then pulling them together after the fact, and having the bandwidth or the amount of oligos you generate in a given amount of time being dependent upon the plates and the synthesizers that you can run through in that amount of time, we use a silicon wafer. Has the same footprint of a 96-well plate, but it's very thin. And we use a microfluidics technology to synthesize our oligos on this silicon plate. Now, we have real estate on this plate that has approximately 8,500 clusters. And in each one of these clusters, we have 121 individual devices. You can consider each one of these devices as a position to synthesize an oligo. So in truth, if you really want to think about it, we make or use 121 microfluidic plates and have 8,500 of them on a single wafer. That results in about 1 million oligos in 24 to 36 hours, and we have 10 to 12 synthesizers. So you're talking about 10 to 12 million oligos that we can generate for any of those previous applications I had shown in the 24 to 36 hour period. Why are we better? Well, in particular with regard to NGS, we have exceptional performance, which comes from using double-stranded DNA probes, which result in very high capture efficiency. That also, as a knock-on effect, has very good uniformity. Actually, I would call it best-in-class uniformity. And we also NGS QC all of our probe libraries before they hit the customer so that we know that what we want is there and the relative ratios are correct. We also have greater flexibility, very easy customization and rapid optimization, but we also offer some off-the-shelf offerings as well, such as our exome. We can scale things to being very small panels like for MRD or very large panels like exomes. And our kit offerings can be modular, or we can give you the complete solution. And finally, the whole idea is that with all of this that we're offering to our customers who want to maximize sequencing efficiency so that you get the most bang for your sequencing dollars off of your high throughput sequencer, your benchtop sequencer, or your in-field sequencer. So I want to go through a little bit of real quick objective metrics. And the way I think of objective metrics is that, if, for example, if you have a gene that has 3 exons, the key is to hit a critical depth of coverage, in this case it's 30x, to make a variant call. Now, understandingly, inherited genetics versus SNP genotyping versus minimal residual disease detection, they're going to have different thresholds. But just for the sake of argument, let's use inherited genetics at 30x. What you want to do is to have all of these exons covered at that minimum depth of coverage. Because if you over-sequence, that's wasted sequencing. If you're under-sequencing a region, well, you could be missing something, which means you have to over-sequence that region, which is going to bring up everything else that's at the appropriate level. And you also want to avoid any off-target sequencing, because all that's doing is eroding the bandwidth available for you to actually generate results with a maximized output on your platform. So what we do with regard to uniform pools leading to uniform capture, I kind of call this my garbage in, garbage out slide. And the reason why is because when we start with a uniform synthesis, this is a chemical reaction. It's not biased by any kind of molecular biology whatsoever, because whether you do your phosphoamidite chemistry in a well or you do it on a silicon plate, it is a chemical reaction. You add a base, you deblock, you add a base, you deblock, wash, rinse, repeat until you get the oligo of length that you want. However, for NGS, this is femtomolar concentrations, and that's not a very large concentration to do an awful lot of downstream applications for target enrichment, so you need to amplify the product. When you amplify, molecular biology and all of the misgivings that come with that, i.e. PCR biases because of GC composition, rears its head. And that's when you have a biased amplification strategy, you can get biased probe representation. Biased probe representation will give you a biased sequencing representation, and this is where you get stochasticity. Twist has a proprietary method, though, where we've been able to maintain highly uniform probe pools post-synthesis so that we do not have this stochasticity or bias, and you get nice uniform coverage across your regions of interest. So the way I think about this is that it all comes down to chemistry. I'm a chemist by education. I like to apply my philosophy of chemistry whenever I can. And the way I think about target enrichment and probe hybridization is that it's really a thermodynamic chemical reaction, which is an equilibrium, where A plus B is equilibrium to C. Well, in our case, genomic DNA is in equilibrium with our probes, and it results in a genomic DNA probe hybrid. Now, in chemistry, you have limiting reagents and you have saturating reagents. And the goal is to make sure that you saturate one reagent so that the limiting reagent is completely utilized and it results in a complete conversion to what you want your product to be. In this case, our limiting reagent is our genomic DNA library. The probes are what you need to saturate, and the goal is to make sure that you saturate them so that you don't ever go back to your reagents, but you always go to your product or your preferred hybridization so you can capture it. Here's the problem. This is all done in an isothermal setting. All of the hybridizations which are occurring are little equilibria reactions because the composition of the probe, the composition of the region of interest, i.e. the GC composition, is going to determine how that equilibrium is going to work. A low GC-rich region has a different kind of equilibrium to a high GC-rich region, and the amount of probes that you need to saturate that equilibria is going to change. So we do have to keep in mind that even though, in a perfect sense, looking at the chemistry, it's a very easy equation, but you have all of these little equilibria reactions competing with one another, and how do you tune each one in an isothermal setting to force complete and uniform capture to give you uniform sequencing results? Well, that's where we go back to how Twist synthesizes and designs their oligos. So what we do is, because we can synthesize 1 million oligos at any given 24 to 36 hour period, what we can do is we can take your design, the regions of interest, and bin them based on the percent GC composition of that region. Doesn't necessarily have to be the same gene or the same region or the same chromosome. It just has to be, what is the GC composition? And for example, I'm going to show in this cartoon, assuming that this is a subset of our silicon wafer, I'm binning them from 20 to 80% at 10 percentage increments. And what I'm showing you is that because we do understand that low GC rich regions are more thermally labile, they have high ATs, they're going to come on, come off, come on, come off, come on, come off very easily at a high or an isothermal temperature, whereas high GC rich regions, they're very sticky. So what we want to do is we want to provide more of the low GC pools in our synthesis to drive the chemistry to completion, because we know they're more thermally labile. But as we get more sticky, we want to add less, because if you add as much in a 1-to-1 ratio as the low end, these high GC rich probes will result in off target, which gets back to those objective metrics of what we're trying to do. We're trying to reduce off target. We're trying to maximize capture uniformity. But the way we do that is stoichiometrically increasing the pool replicates based on composition. And down here, I'd like to give you a kind of a pictorial representation of how we think about this. For example, relative probe concentrations versus GC. Twist has a very large dynamic range of a negative slope because we can synthesize 1 million oligos at a time, whereas our competitors, not so much. But as a result, we can tune these regions such that the relative depth of coverage is flat or uniform across the percent GC composition of those regions. Relative to not having a large dynamic range, you end up getting a positive inflection. You lose regions here at the low GC rich end, and you have sticky regions or high off target at the high ends. And this is where you get stochasticity relative to this highly uniform probe trace that I'm showing you here. And if you don't believe the pictures, then let me give you some real data. Here's the competitor's exome that they weren't able to tune and refine and design the regions relative to GC. And you see this positive slope. This is not what you want to see in a uniform capture and a uniform sequencing run. Whereas Twist, because we have a way of designing, pooling, and synthesizing oligos in a very quick and very scalable, high throughput way, we can tune this capture so that you get nice flat representation across the targeted regions. Another thing that we can do, and I had commented on this very briefly at the beginning, is that we have double-stranded probes. A lot of our vendors use single-stranded probes. They capture and then they have to amplify to use genomic DNA library, to generate the genomic DNA library. Well, as with any standard PCR reaction, if you provide more seed molecules into the start of the reaction, you get more unique molecules at the back end because you don't need to generate a lot of clones. So what we have done is generated double-stranded DNA probes so that we capture both the plus and the minus strand of the region of interest. And what you see is that we generate anywhere from 4 to 6-fold more unique molecules in our capture than our competitors. What does that translate to? Well, Unique library size is inversely proportional to duplicate rate. So if you have more unique molecules, you're going to have a lower duplicate rate or duplicate reads. If you have lower duplicate reads, that means you have more unique reads that you can assign to the analysis that you're trying to do. So that is one of the metrics that we tried to improve at Twist, is that why would you want to waste sequencer bandwidth on duplicate reads which are non-informative? Let's give you those back so that you can either run more samples or run the same number of samples but get more depth out of the results that you have. Another thing that we like talking about is our uniformity, which I've kind of touched on a little bit previously, which is also known as the fold 80 base penalty. So a genomic a whole genome sequencing run has a fold 80 base penalty probably around 1.3 to 1.5. Exome sequencing and target enrichment in general has never quite been able to get to that. And the reason why is because what you have is different places where bias can be introduced, whether it's in genomic amplification of the library, not having uniform probes, which result in low GC rich regions being inefficiently captured and high GC rich regions at capturing too efficiently and or pulling down off target regions. But because Twist is able to tune and modulate the ability of our probes to provide uniform sequencing by tuning the relative stoichiometry of the GC rich regions of interest, we can also tune and lower target enrichment fold 80% penalties on par, if not better, than whole genome sequencing. To get into some of the offerings that we have, and this is what Dr. Paul is going to talk about today, is we do have a core and comprehensive exome. Our core exome is primarily based on the CCDS reference genome. However, our comprehensive exome uses the CCDS and adds additional content that is unique to those references for both RefSeq and GenCode. But at the same time, because Twist can generate custom content very quickly and very uniformly relative to, let's say, a foundational design, we can take the core or the comprehensive exome and we can bolt on different custom contents to meet the needs of the investigator. For example, clinical content or UTRs, or for example, CNV regions. We can tune and balance the content to reflect the needs of the vendor or of the customer. Now, there's a number of ways that we can do this. For example, we can do one-on-one coverage representation so that the foundational design has custom content that's going to be equivalent to it. So for example, let's say that you have intronic regions that you would like to cover equivalent to an inherited backbone of, let's say, the exome. Well, we can synthesize and spike that into a one-to-one ratio. But what happens if there's certain regions that you want to be sequenced greater than a quote-unquote backbone or an inherited region? So for example, let's say you have cancer targets, somatic regions that you want overrepresented because you know that the minor allele frequency variant of those regions could be sub 10%. Well, we can also tune it because we can synthesize millions of oligos in any short amount of time such that you can have a different ratio of your foundation to the custom. On the flip side, we can do the same thing on the reverse. So a lot of customers at Twist are starting to do genotyping by sequencing. So what they do is they add SNP content where it's just a binary call, and they want to have the inherited background be at, let's say, a 30 to 50x coverage, but the SNP calls, they only need 10 to 15x. So why make those regions get covered at the same depth as the other inherited genetic regions when all you need is to make a 1 or 0 call on the SNPs? Here's an example of how we've been able to actually spike in regions and showing that they're uniform relative to the regions we were looking for. For example, here's replicates of our core exome. We wanted these regions as spike-ins. And once we were able to generate them and balance them, lo and behold, here they are with equivalent representation to the foundational set. I'd also like to comment real quickly on our exome and how it's performing against our competitors. Twist offers our exome as a standard 8-plex, but we do also show single-plex data because relative to the vendor that we're talking about or the competitor we're talking about, you could be doing an 8-plex or you can be doing a single-plex. To start, I just want to show very quickly that our singleplex and eightplex data is almost identical. And what we like to say at Twist is you can get singleplex quality data in an eightplex format. What that does is it allows you to increase your laboratory efficiency and your bandwidth such that you can, per every hybridization, get 8 samples instead of 1. Relative to some of our competitors that do 8 plexes, pretty equivalent at the 20x, but not necessarily at the 30x. And if you were to look at 8-plex versus singleplex, or 8-plex or 1-plex versus singleplex, I think the data speaks for itself. So the idea is that you can get up to 2 times more samples per run when you start thinking about how we are able to uniformly capture complex libraries in a diverse hybridization reaction in an 8-plex solution, but not have to take the performance hit relative to a singleplex. One of the things that Twist excels at in the NGS market is custom design. We've been doing it since we started in 2017, and we continue to learn and improve our in silico tools. So we have come up with this very rapid design, build, test, learn, or DBTL system, or what I like to call the learning loop, where a customer will come to us with regions of interest, we design it, we build it for them, give it back to the customer, they test, we all learn from it, and if we have to, we'll do a redesign. The design takes anywhere from 24 to 72 hours. It is free of charge. You give us your regions, we'll design it for you. We can synthesize it for you in about 3 weeks, and then it's up to you to run that, learn from it, share your results, and then we go back through this. The key here is that 2 things with regard to the learning loop. 1, you want to get through it fast. You don't want to be bogged down in the learning loop. And secondly, you want to spool out of it as fast as possible. You don't want to be constantly spinning your wheels, so to speak, in the learning loop. So what we have found is that a lot of customers have been able to go through the loop once, maybe twice, 3 times if they're trying to get marginal gains, and then they've been able to spool out of it and scale and execute in the market. So if you think about a 3-week turnaround time from design to build, and then let's say you're testing and learning in another 2 weeks, we're talking about possibly iterations which are happening in a month to a month and a half. You could get 2 iterations in the time that maybe with some of our competitors, you're just getting your probes. And as we all know, time is money, time is results. So once again, getting accurate and high fidelity results in a quick amount of time is going to save an awful lot of angst. Here's an example of how we went from an initial design to a rebalanced design with an N of 1. This is 1 time through the learn. Actually, I guess I would say we went through the loop once and then we learned once, and this is the result that we got. So the initial design, once again, you don't want to see a positive slope, but with 1 iteration, we were able to flatten that out with an R-square of almost 0. This is what happens when we use the learning loop. And what we have also found is the more that we've improved our design tool, in a lot of cases, some of our initial designs look more like the right rebalanced design than in our previous iterations where we were getting good, but absolutely not the greatest that you can with these custom designs. So to kind of close out, I would like to say that once again, we have very diversified and very modular kits that we offer with regard to generating your genomic DNA libraries, adding the adapters, whether we're doing the enrichment of these libraries in 1 day or within a day. We offer catalog and custom panels. So just to think that we can give you the full solution, but at the same time, we can give you the a la carte as well to what meets your needs. So in closing, I know that we like to say that you can pull any lever that you want. You know, the idea of flexibility is being able to turn on or turn off multiple switches. I think Twist has been able to place themselves in a very enviable position where we don't think about how we should turn off or turn on multiple switches to meet a customer's need, because I think we also know that it's not black and white. We deal in shades of gray. And so the way I like to position Twist is that we don't pull levers anymore. we modulate, we turn dials, we allow you to keep everything on, but maybe just not on as a completely, blinding light. We can make sure that the things that you feel are needed, the things that you think that are wanted, the things that need to be improved, we'll make sure that we work with you to modulate what is required to get you to the place that you need to be. So in closing, what I would like to say is one of the dials that that Twist feels that they've been able to do is with this collaboration with SOPHiA. So let's think about it this way. This is how I believe that, this is how I look at it, and this is how I know that Twist looks at it. You get to get this superior uniformity of target enrichment with Twist. And then you add in the informatics tools and algorithms to help optimize, utilize, drill down, and refine the data that you can only get with a highly uniform capture. And the partnership results in this wonderful copy number variation detection methodology that has been, up to this point, not quite been able to be tapped into. And I think that this partnership between Twist and SOPHiA is kind of a marriage that allows for investigators to really take their CNV calling using NGS to the next level. So with that, as my CEO would always like to say, if you don't believe us, we dare you to compare. And so by all means, feel free to contact us, kick the tires, tell us what you think. And I think with that, I would like to transition to Dr. Emily Paul of SOPHiA Genetics so that she can show you how she's utilizing a uniform exome in the strategies of accurate and reliable CNV calling with SOPHiA Genetics. So Dr. Paul, here you go.
Mark, thank you so much for that. And thanks for that great review of what you guys have been doing. It's really quite exciting to see. So I'm going to go ahead and share my screen here. All right. So thank you again, Mark, for your introduction. And I'm exactly going to talk about that coverage uniformity. You mentioned that great way that we're able to look at all of the exomes. So thank you for that lead in. Just a note to say that the SOPHiA platform is currently intended for research use only. And we have a lot of different notes and information that we can share with you in that regard. Now, SOPHiA Platform is a multimodal platform bringing together information from different spheres of the holistic individual, including genomics, radiomics, and presenting them together to account for each of them. As you can imagine, today we're going to be largely focusing on our genomic series and what we can do there, particularly for copy number variations. Now, as I mentioned, SOPHiA has a lot of different components to its platform, and the idea of this is to have a universal platform. The universal platform allows us to understand what data looks like, what all sorts of data looks like, whether the sequencer is different, whether the type of panel that you're running, whether it's a targeted panel or an exome, and really be able to appreciate the biases that this actually introduces into the data set itself. And then we can take it from there to identify the variants and provide them in the platform itself. Now, we're talking whole exome sequencing, and this is a great thing to talk about. I think it's an incredibly useful tool, but there are pros and cons to it, as with running any type of assay you may run. Now, the obvious pro to this is that there are more genes. That's great, more data. And in the very American saying, the bigger the better, right? We're getting all of this information and trying to use it. And that means we can see new variants, see new combinations, understand those novel variants related to disease. And we're also able to streamline a lab workflow by collecting all of this information at once, by allowing labs to develop one assay as opposed to multiple assays. But on the contrary, we also have some cons that we have to consider. And one of those is the exact opposite of the first one. There is more data. There's just more of it. And while there's the possible power of higher understanding, you have to be able to actually process that data. And there's also challenges when identifying the variants there within. In particular, copy number variation detection in exome-based assays has been difficult because you cannot achieve the resolution you would see in a targeted panel. And then finally, you have to be able to filter all through this information. You have to be able to process this. You have to be able to see what you should and can see. And then you have to be able to put the blinders on as appropriate. And the SOPHiA platform is able to do this with you. Now, we're looking here today at over 21,000 genes. So again, bigger data, bigger power, but also bigger concerns to be able to actually get to the root of the information. And so with Twist, we're able to take a look at it through data upload, secondary analysis, and tertiary analysis. And I'm going to talk to you about each of these 3 different targets here that you'll see as 1, 2, and 3. And we'll dive into them as we go through our talk today. Now, as I mentioned, the first part is how do you get to the data? Right? You can generate the data, but how do you access it? And that's actually through a streamlined workflow that we provide at SOPHiA. So typically those in the SOPHiA community are healthcare institutions, and they're able to generate their exome-based data. They've processed their data, they've processed their samples, they've generated raw genomic data, and they're able to upload it directly into the SOPHiA platform. So there's no middleman. There's just a straight upload of those FASTQ files right into the platform, where variants can then be identified, annotated, and then pre-classified. And that pre-classified is part of SOPHiA's signature to help, again, filter through large sets of data. And this allows individual experts to analyze the data, but also to generate different types of reports to be able to produce a quantifiable metric out of this 21,000 gene panel. And of course, something that we take incredibly seriously is privacy and security. In fact, we have one of our ISO certificates in this, and it allows us to be confident when working in a large scale data set that needs the support of cloud processing. But we also want to give those institutions the confidence and the security that they demand. Now we're coming into that second part, that secondary analysis. And sure, we can talk about SNVs and indels, but today's all about those CNVs, or those copy number variations, and how we detect those with confidence. And it's incredibly important to be able to detect these with confidence. You know, we think of them as an extra part of an NGS assay, but the reality is being able to detect CNVs in exome-based NGS applications can greatly increase your diagnostic yield by approximately 35%. And this is a big value that can come into work in different types of labs. But there's also different challenges that come hand in hand with the detection of copy number variations. And some of those challenges include that typically lower coverage that we see with larger panels. The bigger the panel, the lower the coverage overall. How does that actually affect our ability to call copy number variations? And then some regions may not be as well characterized, so we may not understand them. You know, we have those compact panels where every gene is perfectly clear to us and what it can actually then do. But then as we get into these novel regions, these novel targets that we talked about as a benefit of running exomes, there's still some lack of clarity there. And we look to provide a resolution to that. And as expected, with larger panels comes larger levels of noise. So we have to be conscientious of that, particularly for copy number variation detection. Now, in order to be able to process this, I just want to note some requirements of CNV detection in exome-based applications. And these will come into play as we talk a little bit more about how we process this data. And so we look to have at least 8 samples. They should be prepped in parallel in the same lab, preferably sequenced on the same run, but they can be sequenced on different runs as appropriate. And then the samples are actually compared to each other and coverage is normalized. And that's a point we're really going to dig into. And then finally, we want to know what are problematic regions. What are regions that could prevent us from clearly understanding the copy number variations there within the assay. Now, let's go into the idea of the samples and how we detect those copy number variations. The assumption is that the CNVs are unlikely to be shared by a significant number of samples in the run. We understand there are exceptions to that, but as we talk to different groups, we can understand how they load their runs, how they process their data, and help them tailor their assay to what we're going to see. Now, one thing Mark mentioned was the idea of coverage uniformity, and that is so crucial to being able to understand what the profile of a region should look like. So here on the left-hand side of the screen, we can actually see a reference genome with exon A and exon B, and looking at different samples. But here in sample 1 at exon B, we see a decrease in the lack of coverage due to a deletion. Now, because we have a uniformity of coverage over the entire assay, we're actually able to say that that region is in fact a deletion, and we're able to process that information for an exome. Now, if we look at the second plot on the right-hand side of the screen, we can see that there is a possibility of much greater noise. Now, what do we think about this noise, and how does it affect CNV calling? Well, noise is introduced in so many steps of any molecular biology assay, going from the DNA extraction itself all the way to the processing and sequencing as well. So if we think about the different places that the noise does in fact come in, what we must do is minimize those regions as much as possible. So as I mentioned earlier, we look to have those 8 samples in a run, because it helps us understand the noise profile of not only that capture, but the sequencing itself. And it allows us to be able to look continuously over the assay. Now, I've talked about noise and where it generates and how different labs can introduce that into their assay. But what we can see is in an assay where the residual noise level is less than 10%, we're able to confidently call a 3 exon deletion here in the BRCA1 gene. We see a continuous profile, and these are SOPHiA what we call dot plots, and we're able to look at the gray dots in the background as the normalization of coverage, and the blue dots as the no copy number variation. But now as we start to increase that noise, where we're starting to see it creep up to about 15%, but most at 10%, we're getting into medium, we're starting to see these discrepancies. We're starting to see the gray expand as well as the blue, because that normalization is changing. In fact, we can start to see quite a big spread over the classic 2 copy number. And finally, when we get greater than 15% noise, you can in fact see quite a few X's throughout this display here on the left, and that's showing rejected regions. So as the noise gets greater, it becomes much more difficult to be able to identify copy number variations in a confident manner. And I emphasize that confidence because you could potentially say, it looks like this, but how do you confidently know what you're looking at? And here we can see a zoom in on some of these regions. And we can see one sample in particular from a batch of 23 that happened to be processed. Again, we see those gray dots in the background, and we see that representation of a heterozygous deletion while we see nice clusters of our reference samples. Now, how do we go about this? Well, let's take a look at a specific region using some IGV screenshots to be able to zoom in on what we're seeing. We're going to be looking at this hemizygous deletion. We'll see it right here over these 3 exons. And the way that we can actually see this is to look at the normalization in 2 manners. First, we're going to look at the normalization of the samples. Remember, I mentioned the noise of the processing of samples actually gets included. But then we're also going to look by region itself. And those 2 normalizations are actually quite important to be able to confidently go forward here. Now, what we're looking at is still that screenshot, but of course, across the top, we see this pink line inserted to be able to identify a normalization baseline. And we are able to normalize across this whole region, across the samples as well. So in fact, the algorithm detects from those 8 samples that are processed together, which ones work as the best reference samples for that run. And here they're largely looking at coverage for the assay itself. Now we're going to take those reference samples that we set, and then we're going to be able to target and look at the CNV detection through a hidden Markov model and be able to identify them. And then once the results are provided, we're also then able to dig into confidence levels. Because as I mentioned, it's nice to be able to call these, but we have to think to our confidence levels for identification. And it's important to think of the different regions that we can see, because this also applies to very long regions. You know, we have to consider both the small events, the exon-based events, but we want to be able to detect those longer range copy number variations. And we can see an example here, both in the IGV screenshot noted above, where we're looking at greater than 54 exons. And then we see that actually presented down here where it's continuous. And we're seeing that range to really get into what we have. Now, unsurprisingly, there's higher coverage and more points of information that are coming together to make this approach, to make this visualization. So when we think about regions, we're not just thinking about the one probe regions, we're actually thinking about a greater region itself to be able to really appreciate what is visualized there. And with that higher coverage, we're able to have higher confidence in the CNVs that we're calling. Now, there are quite a few long-range events, and as I mentioned, we want to have those more probes, those greater length of probes and that greater information of continuous information, in fact, to be able to identify those copy number variations as we go through this. But we have a way to look at these, and the shorter range events are not neglected, because we have the ability to look at different probes and to be able to realign what we're looking at to confidently call those as copy number variations. Now, all of this is to say, copy number variations are part of an equation. We mentioned at the beginning, secondary analysis includes SNVs and indels, but that CNV should not be left out and should be encompassed in the assay that you're running so that you can have a streamlined workflow. Now, how did we go about this? In fact, there's approximately 187,000 regions that are assessed for copy number variation out of those 21,000 genes that we noted. And here you can see that we looked at quite a few benchmarking ways to appreciate this. And in fact, our development for the model and algorithm that calls copy number variations was developed from one of those targeted panels. You know, we were able to look at MLPA confirmed data using our hereditary cancer solution, but then we were able to extract that and confirm it within the twist assay for the exome. So we're able to take that information to understand what continuity across regions we're able to see. And we're also able to look at the number of fragments required to do this. So we're looking at 160 million paired end reads. So we're looking at 80 fragments total in this situation. We can start to see the length and the length of the regions that we can get into. Now, as you would expect, the higher the coverage, the greater the resolution. But even as we get down into some of these lower numbers, we're able to still have a clear number of copy numbers called over a small region. And even as we get into some of the greater length, so we could see length of region here, even as we get into this, we're seeing this consistently throughout the assay. Now, we talked about uniformity of coverage both here and in Mark's talk as well, but we're able to look at coverage uniformity in the SOPHiA context. And here we see the median coverage normalized to 0, and it's represented on this plot across all the chromosomes. We're seeing that consistency that allows us to be able to write these algorithms to detect the copy number variations because of that uniformity of coverage throughout the assay. And how does this look? Well, actually, in fact, the sensitivity is greater than 90% if we're looking at the 40 million reads. And here we can see the pairs or the read fragments. And we're seeing the number of consecutive regions starts at 1, but that confidence and sensitivity goes skyrocketing the greater number of reads that you see through the assay. And just one note to talk about tertiary. I know we focused much of our time on that secondary, but we do have some mechanisms within the SOPHiA platform to be able to understand how we can filter this data. So thinking about that 187,000 regions that are covered by copy number variations and the full set of 21,000 genes that we're looking at, we really need to be able to pare down what we can see and what we can appreciate so that we actually don't get lost in those cons that we've mentioned up front, so that we're able to focus on the benefits. So the pre-classification that we do within SOPHiA allows us to focus the variants that you look at. We're able to apply bioinformatic virtual panels, so you can focus either on phenotypes, on known gene lists, whatever is appropriate for your work. Then you can trim those to rare and functional variants, and if appropriate, apply a familial variant analysis to your work. So here we have those all accompanied, are all put into the SOPHiA platform. And with that, you can actually make a report that allows you to take that information and share it with those around you. Now, there's one other component of the SOPHiA platform that we didn't get a chance to talk about, but we have a SOPHiA community of users who allow us to contribute information to each other. Now, while we understand there's individual experts and great prolific understanders of genomic modifications, it's hard to be an expert in everything. So why not rely on the community of those around you for insights and information that can help you better understand those rare disease variants that we talked about up front? Why not allow those who have the knowledge to support you? And in that sense, we think of the idea, two minds are better than one. Well, here we go up to greater than 1,000 members of the SOPHiA community, allowing us to push forward into what we hope defines exome studies. Now, just as a quick summary, we look at the Twist Comprehensive Exome and know that you could process the data however you'd like. You're able to go in there and manipulate it, play around with it, and really poke all the holes in it so that you can interpret what you want to see. We have advanced analytical performance of detection of SNVs, indels, and copy number variations, and I hope I've shown you how we're able to get down into sensitive CNV calling. In fact, it goes down to exon-level resolution. And then the tertiary tools that can actually support that. And I'll go and say thank you and ask for any questions that you may have, and also thank our colleagues over at Twist for allowing us to talk today.
Emily, thank you so much. That was a fantastic presentation. And Mark, thank you too. All right, so I think we're going to start taking a few questions, and they're coming in via the Q&A box. So thank you, everyone. So before I go to our audience questions, I wondered if I might just ask you guys. I mean, I know from working at Twist, we're very much focused on really enabling our customers, and this really comes across in the SOPHiA presentation as well, especially with your brief mention at the end of the community. So I wondered if I could just ask the two of you, what are you excited about with respect to this relationship here and the benefits to our customers? Emily, perhaps you want to go first.
Yeah, and then happily. In fact, I'm incredibly thrilled about the Twist partnership, because I've seen I've seen you guys at conferences and what you're able to do in building your probes and your library bases. But it's really that coverage uniformity that will actually allow us to synergistically work together. And I look forward to being able to get better resolution, to define new variants, to see novel disease-causing variants as we go about our partnership.
I think the thing that I can speak from the Twist side is that, I think CNV has been one of those methodologies or those analysis tools that has not really been able to break through with the NGS community, because you always see stochasticity in coverage for regions of interest. Twist now has a way to make that, as Dr. Paul said, have a very uniform capture. But even so, those customers, those clients that we want to expose this to, they may not have the informatic tools, or they may not have the in silico wherewithal to make that happen and provide it in a reported setting for, let's say, for a customer. And so what I think is that when you pair, once again, this very highly uniform methodology with, which is Twist, but now take the expertise of SOPHiA in understanding how to drill into the data, you just enable customers. So it's not like Twist could do it without SOPHiA, and I don't think SOPHiA can do it without Twist. I think this is where everything comes together. It's the perfect synergy.
Agreed.
And what kind of sort of applications and samples do you see people working with on this? So Emily, you've been very clear that obviously at this time it's a research use only product, but are we talking biopsy samples or liquid biopsies, or are we talking you see it being used in oncology research or fetal sort of abnormality research, or what are the sort of clear applications for this stuff?
Sure, we're actually seeing quite a few applications, excuse me, for this, and it's actually covering the gamut that you mentioned there, because we're seeing a greater push to bigger data in all avenues. Originally, where we saw those targeted panels for somatic applications in oncology and really focusing on specific mutations, but that curiosity that we all possess knows no bounds and wants to see much more data to better understand and better profile what individuals are going through in oncology. But it's unsurprising to see that expansion as well, to push into fetal medicine and to understand those rare diseases in pediatrics.
Okay. All right, well, I have more questions, but I want to dig into a few from the audience, actually. So I will. So, Mark, to begin with, there's one coming in about panel design. So specifically asking the question, often we see a GC-rich exon 1, which can cause, coverage issues. So is this adjustment that we're making for the high GC content taken too far in these instances of GC-rich exon 1?
No, actually. So there's another slide that I would love to have shown, and I didn't think about it in this regard. But if you were to actually show coverage of our exome relative to GC-rich regions, some of our competitors at the high GC-rich region end up having nothing, and then what they do is then they get a spike, and then it starts the coverage uniformity plot starts to take the sigmoidal representation that you would expect. With Twist, it just starts there. So we know how to tune the design such that we can get those GC-rich regions, but we're almost suppressing the capture so that we're not over-representing those regions to the point of having what I like to call targeted poisoning. The probes are so sticky that they'll find anything similar throughout the genome that could also be captured. So I call it target enrichment poisoning. And that's why people, or some of our vendors, try and avoid those regions, because they would rather have a missed region rather than have bad data or high off target. And at Twist, we like to say we won't miss anything. That there's no region that left behind. Now, I will say this as a caveat. If you can't sequence it and align it in a whole genome, working with Twist is not going to help you, okay? Because sometimes the region is too repetitive, too non-unique, signs, lines, ALUs, STRs, etc., that the alignment tool just won't work. So target enrichment will not help whole genome make it better. So that's the one caveat I want to say.
Good to know. Well, actually, so that was a question from Tobias. So thank you for that. But on a related note, a question from Pierre Giorgio Modena. And I think this is one for you, actually, Emily. So it's with respect to using the 8 samples reference. So in a set of, say, 24 samples, will the 8 samples reference set be fixed, or will the algorithm apply permutation testing? So, will you have a permutated set of reference samples?
So a 24 is actually, in fact, much better for anything. We say a minimum of 8, but if we look to a larger set of data, we have a better understanding of consistent coverage profiles, and we can look to them to see which are the best 8 to apply for this modeling. So we will see that the flexibility in the normalization to be able to get the most correct baseline.
And if I could just slip in a quick question here. So
Please.
you showed nicely in your data set the loss of heterozygosity, especially with the lower noise. But can we also see gains of heterozygosity? Would you see it in a stepwise manner, or where, to what degree of resolution can you see once you start to see gains of?
Yeah, so we can definitely see the 3, the 4 copy numbers as we focus really in the germline avenue for this presentation on copy number variations, but we have algorithms that allow our somatic modeling to look at more fine-tune resolution in sense. So if you have 4.5 copies or 22.5 copies, right? We're able to actually determine those as well. But here in a germline exome, we really do see 0, 1, 2, 3, and 4 as appropriate.
For those of our customers working with HeLa cells, it could be any number of copies. Right.
Favorite number, yes, absolutely.
So another question, which coming in from Margarita: any recommendation for designing a panel to detect CNVs in custom panels? So would it be recommended to cover, I think what we're saying here is a larger region than the gene of interest?
So sometimes, sorry.
Or Emily, I was going to say, probably a question for either of you there. So Emily.
Well, we could both give it a quick response.
Yeah.
We see the expansion of regions a little bit into those intronic regions very helpful. So when we're thinking coding regions, we do have a little bit of overhang in our hopes for design. And as we mentioned, the greater number of probes, the better resolution that we're able to see.
I would comment on that as well. I think the de facto methodology for just mutation detection in coding regions is that we design our probes inside the margins of the coding region. But when you start to have a nuanced design like CNV, having a little bit of bleed into the intronic region is essential. So it becomes almost taking a standard design and then adding almost an overlay of a custom design that is CNV specific. I also like to see with CNVs that if you think you know that a certain gene or a certain region is going to be maybe holistically or partially deleted, I like even going outside of the UTRs, because you kind of want to see, do I get an idea of where the breakpoint is? Because if all you're doing is coding region, you can have a coding desert in between 2 adjacent genes. And if that next adjacent gene, either 5' or 3' is normal, where the heck did the break occur? And so I like bleeding out a little bit, even on the extreme ends. Sometimes I would say maybe 250 kb, just to give you a little bit of flank. And those can be just simple tiled probes. You don't need ultra tiled probes in those regions.
Okay. Question from Veronique. What is the rate of mistakes in finding CNV for NGS of target genes? So she understands that it's possible if the coverage is not good enough, but is it possible to find a complete gene deletion or insertion in a good coverage profile that could be false? So I guess the question there being false positives, false negatives. What do you have any comments on that one, Emily?
Sure. So as with any part of any exome assay, right? We're going to see the possibility for false positives and false negatives. But as we developed this assay and really looked to orthogonally confirm CNVs, to be confident in how we develop this, we're able to actually be able to zoom in, to target what we look at and minimize those. Now, we do still see some labs that look for orthogonal confirmation of their MLP from their MLPA assays or whatever they may run. But we also see the confidence growing in our labs that run exome-based solutions, where they are less likely to do that orthogonal method because they have the confirmation. So of course it is always possible, but typically this goes back to that idea of confidence that I mentioned. We will reject regions rather than call them with a false positive or a false negative.
Okay. We are coming up on time, but as we've got a few more questions, I'm just going to carry on a little bit. I hope that's all right with you guys. Just a couple of sort of slightly technical questions. So from an anonymous attendee, do you follow standard VCF formatting for reporting CNV calling?
So we're actually expanding the way we look at our CNV annotation, and we're constantly changing our platform to meet the needs of the market. So right now we have different ways to take the CNV data out of the platform, in addition to the reporting format that SOPHiA provides.
And then I believe you mentioned this actually in one of your last slides, but it's been asked a few times, so I'll give you a chance just to re-emphasize this point. Can SOPHiA detect single exon duplications?
In fact, we can. We're very proud of having that noise resolution and dampening to be able to get to that single exon. Now, of course, it depends upon the depth of coverage that you sequence to, and the size of the exome itself. You know, we want to take that into account and how long it is and the clarity we're able to achieve. But yes, our resolution is down to a single exon.
Mark, one of your final slides was dare to compare. So there's a question in the chat actually asking, what's the main difference between Twist probe chemistry and one of our competitors? So would you have any comments or to make at this point about whether they should try out Twist chemistry as well?
When you say chemistry, do you mean like the library construction chemistry or the methodology of synthesizing the probes?
Well, this person's recently bought an exome kit from one of our competitors, and they're looking for a reason to spend on Twist chemistry too.
Once again, all those slides that I wish that I wouldn't have removed from the presentation. So I think the thing that I can come back saying with regard to the Twist solution is that we've spent the better part of the past 3 years refining the different aspects of it. For example, the diversity of the way we can generate the libraries, whether it's mechanical or enzymatic, using UDIs instead of CDIs so as to minimize adapter dimers and give more unique molecules. The idea of using double-stranded probes, which allows us to generate more unique molecules in hybridization. I mean, the key is that the more unique molecules that you take into hybridization, the lower the duplicate rate is going to be, so you're not going to waste sequencing reads on non-informative regions. The other thing is by taking our probes and having them uniformly synthesized, not uniformly pooled after synthesis, allows us to control the process up front where it's most important, rather than providing it to a robot or to a human to make sure that they stoichiometrically mix it after the fact and get it right. I mean, the fact is that we're doing it up front rather than on the back end. So I would say that in and of itself provides us with an awful lot of avenues to take that product and make sure that it's as uniform as possible so that you're not wasting any sequencing. And with the data that I've shown already, I mean, a reduction in duplicates by 8% and a fold 80 increase of 0.2 to 0.5, I mean, that's two times more samples. So I would take that Pepsi challenge, and I would ask that person to take that Pepsi challenge. I'd be happy to stand by it.
Well, Rodrigo, I hope that gives you a suitable material to make the case where you are. I will just answer one final question then, which is again from Rodrigo, and then we'll call it there. So what would be a good way to get inversion with a custom design panel? So in this case, like Boland mutation in the MSH2 gene, would this be possible?
I'm going to let Emily talk about this one a little bit, and then I have an idea too.
Yeah, absolutely. Chime in about the probe design, but I'll speak bioinformatically that we're able to detect inversions like the Boland, ever so popular in hereditary cancer testing, because we're able to look at qualified and known breakpoints for that particular mutation. So I would say one of the limitations is, in fact, having identified breakpoints. And of course, positive controls are always nice to help. But there are some known positive Bolands where we've been able to detect them. Mark?
So I would say that in combination, once again, with having the informatics tools able to recognize it as an inversion, then is, okay, making sure that you can see it on the sequencing read side. So inversions, complex functional changes are hard because in some cases, no one knows where the breakpoints are. If you do, let's say, for example, you know where the breakpoint is even on one side, what you will do is you're going to probably generate probes that are highly tiled so that you can walk the probe across the possible breakpoint to account for any and all possible variation of that. And then what I like doing is I like taking and putting a probe that's absolutely known to be associated with a non-breakpoint region, and then also on the inside of the founder gene as well. You can do it on the opposite side as well. But here's the thing. If you highly tile the probes, you allow the inversion breakpoint to be walked or expand or contract. At the same time, what I would also say to someone, if you're really looking for inversions like that, generate fragments that are larger. Do a larger insert size and do longer reads, because that's going to allow you to have a better possibility of sequencing across the breakpoint with these multiple probes. And then the reads, even with the probes being slightly off, is just going to be confirmation of the break. It's a combination of informatics design and sequencing strategies on the bench. You got to do it all.
Right. And Mark, I need a yes/no answer for this one. If someone needs help with custom panel design, can Twist help them?
Heck yeah.
An
emphatic yes. Yes.
All right. Well, hopefully that answers as many questions as we have time for today. I'm sorry if we didn't get time for your questions. If you put your name in the Q&A box, we will try to follow up by email. And as you've seen, I hope from Emily and Mark's talks today, we're here to help. We have experts in-house, and we'd love to support your various research and experimental efforts. So please do reach out and contact us. A reminder, when we end the webinar, you're going to get chucked onto a quick survey. We'd love to hear your feedback, and please don't hesitate to contact us if you have any further questions, and we'll hopefully see you at the next webinar. Thank you so much.